All right, so today I'm going to talk about the two processes to create ATP in a cell. The two processes that we're going to talk about are respiration and fermentation. The first one, respiration, can be split into two different categories, which are anaerobic and aerobic. The way the respiration process goes is that we're taking a glucose molecule and we're going to go ahead and break this down through the process of glycolysis. From here, these broken down molecules are going to go ahead and go through the TCA cycle, or better known as the Krebs cycle. And from the Krebs cycle, they're taken to the electron transport chain, which will create a proton motive force. And what this means is that ATP can be produced through this, through the process of oxidative phosphorylation. And that's how respiration is going to go ahead and create ATP. Now, with respiration, like I mentioned, there are two categories. And the first one, aerobic, what it does is its transporter is oxygen. It utilizes oxygen as its transporter to move those electrons around to undergo this process. Now, anaerobic, because we don't have that oxygen present, we're going to be using a different molecule, and that could be nitrate or any other thing that's in that cell at the time. And then the other process that we're going to talk about is fermentation. Fermentation is a little bit different because we don't have that oxygen present necessarily. And it's also more so that if in this process you have to move electrons from one organic compound to the other. So with this one, we're going to start with the same molecule, glucose, and we're still going to go into the same process of glycolysis, but instead of using oxygen or a nitrate, these broken down electrons, molecules, are going to be taken by a transporter, NADH, and they're going to transfer them to either pyruvate or a derivative for transfer. And from here, once they're transferred, this is actually going to regenerate NAD positive. Now, why you choose one over the other is because respiration actually creates a higher yield, where the theoretical maximum would be 38, and fermentation is going to give you a theoretical maximum of 2. And that's all. Thanks.